Okay, looks like we're going. So what I want to do is go over this particular paper called Distant Sequence Similarity Between Hepcidin and the Novel Coronavirus Spike Glycoprotein, a potential hint at the possibility of local iron dysregulation in COVID-19. So uh, I wrote a different paper, or at least an essay about the subject, where I personally believe that the not just this particular coronavirus, but a lot of viruses in general, try to find ways to dysregulate your mineral concentration, uh, I should say metabolic pathways in your body in order to free up those resources for their own enzymes. So a lot of enzymes and proteins require mineral or metal cofactors for them to properly fold or to properly function. And in the my belief is that iron ions are playing an important role for the viral machinery to function correctly, on top of a bunch of other minerals. But what is particularly important for iron regulation is that iron in your body is very, very tightly controlled because it's a very toxic substance when it's unbound. You can generate free radicals if you have unbound iron in your body. And also, what was it? Yeah, free radicals can be generated, and also the iron itself is reactive, and iron can cause blood clotting if it's not properly bound. So there are many uh, proteins that your body produces in order to make sure that iron is sequestered and packaged so it's not doing damage to the body. So, and this is tightly regulated through hepcidin, is one of the major peptide hormones that regulates how iron is moved throughout the body. So one would argue that it's probably very advantageous for a virus to basically hack into that regulatory mechanism to liberate some of the iron for itself. Okay, so I read a different paper where they did some bioinformatics and they inferred that there are probably some of the open reading frame proteins that are produced by the virus are actually able to attack hemoglobin directly and be able to liberate some of the iron from the hemoglobin. But that is a completely different paper. I believe it did end up getting published, but I'm not going to focus on that for now. I'll probably bring it up in a later video. But right now, we're going to be talking about how this particular coronavirus might be able to hijack your iron metabolism using at least a small amino acid sequence that looks very similar to the hepcidin peptide hormone. So let's jump into it. The spike glycoprotein of the SARS-CoV-2 virus, which causes COVID-19, has attracted attention for its vaccine potential and binding capacity to the host cell surface receptors. Much of this research focus has centered on the ectodomain of the spike protein. So you have your coronavirus, and then you have your spike proteins that are coming off of it. And when they talk about the ectodomain of the spike protein, they talk about the part of the spike that is on the outside of the virus, the ectodomain, the outer domain. The ectodomain is anchored to a transmembrane region. So transmembrane region, remember this is an enveloped virus, so it has a cellular membrane that it stole from its host. And the transmembrane region is a part of the protein that goes through the membrane. And then followed by a cytoplasmic tail. So remember, the cytoplasm is the inside of the cell, the cytoplasm. So your spike protein has its ectodomain, it has the part that goes in between the cell membrane or within the cell membrane, and then it has a little tail that's in the cytosol of the cell, and then also in the interior of the virus. Here we report a distant sequence similarity between the cysteine-rich cytoplasmic tail of the coronavirus spike protein and the hepcidin protein that is found in humans and other vertebrates. Hepcidin is thought to be a key regulator of iron metabolism in humans. So I read somewhere a good analogy is insulin works on glucose. Hepcidin works on iron. So iron is fulfilling the same role in iron metabolism as insulin does in glucose and blood sugar metabolism. Um, it's probably not exactly correct, or definitely isn't exactly correct, but that is one of the ways that you can remember how important of a role this hepcidin peptide is. So, continuing, an implication of this preliminary observation is to suggest a potential route of investigation in COVID-19 research field, making use of an already established literature on the interplay of local and systemic iron regulation, respiratory infections, and the hepcidin protein. The question of possible homology and an evolutionary connection between the viral spike protein and hepcidin is not assessed in this report. So here we go, the main text. As of the date of this paper, 175 countries and regions are tackling the challenge of the pandemic caused by the novel coronavirus. Coronaviruses, first described in the 1960s, are mostly present in birds and mammals, and there has thus far been 7 coronavirus infectious disease outbreaks in humans causing respiratory illnesses. The severe acute respiratory syndrome coronavirus SARS-CoV in 2002, MERS, and now the SARS-CoV-2 of 2019 are 3 of the 7 known coronavirus infections causing severe human disease. So those are the severe ones, where you get a huge viral load, and they really find a good way to take advantage of your receptors and your proteases to get into the cell, more so than the other 4. Continuing, this positive sense single-strand RNA virus. So RNA, you can have DNA and RNA viruses, and this one is a single-stranded RNA, so there's only one strand of RNA that is within the virus. And positive sense means that the ribosome or your cellular machinery are able to start translating the RNA directly into proteins. That's just what positive sense means. This positive sense single-stranded RNA virus family possesses the structural protein spike, or S, so you'll probably see S written in the paper a lot, the membrane and envelope proteins, along with the nucleocaspid protein. It also has the largest genome among RNA viruses, so coronaviruses are just large viruses in general for RNA viruses. Much research interest is devoted to the spike glycoprotein, forming the characteristic corona, hence the name coronavirus, and its importance in the development of vaccines and antivirals. So they focus on the spike glycoprotein because, since that's the part of the virus that is exposed, then if you can create antibodies to those exposed parts, those are going to most likely be antigenically favorable. There are some reasons why you might not want to make antibodies against it, such as autoimmune diseases and immune dysregulation, but that at least to attack the virus, that's what would be used, or especially for vaccine development, we'd be using the epitopes or the structure of the spike protein to be making the vaccines. The angiotensin converting enzyme 2, or ACE2, is thought to be its receptor on host cells, and we're pretty certain about that. The spike protein is formed of a receptor binding subunit, or S1, a membrane fusion subunit, S2, and a single pass transmembrane domain, and a cytoplasmic intracellular tail, or CT. Of note, the S1 domain has a similar fold as human galectins. Oh, really? Galactose binding lectins. I'll have to look into that. Lastly, in terms of the putative primary function of the spike protein, Lee comments that because coronaviruses must enter cells for replication, membrane fusion is a central function of the coronavirus spikes. So, a basic question that might arise is, what exactly makes the pathobiology and disease course of these particular viruses unique? And could it be that, in addition to viral replication inside of the particular type of human host cell, Other intracellular processes specific to these viruses are involved. So I don't know if they're referring to coronaviruses explicitly or these three coronaviruses, so SARS, MERS, and SARS-CoV-2, how they are different from the other four coronaviruses that are much less lethal. I'll go over it later. So having this in mind, we wondered if there might be any sequence similarity, and thereby potentially structural similarity between the SARS-CoV-2 spike protein, which has that many amino acids, and any vertebrate proteins. So, excuse me, they did something similar to what I did earlier, which is where they do a sequence alignment, and you can see if there are any regions or areas where there's a lot of homology between what the spike protein and certain amino acids that are in some of your peptide hormones. That's what I did, and that's what these people did as well. A simple blast search does not reveal any similarities with human proteins, and I will have to disagree with that because I was able to do some blasts as well, but I think you need to be smart with it by toggling the different types of you need to toggle what spike proteins you are putting into your blast search, and you also need to toggle your the proteins that you are aligning with the spike proteins, the human proteins that you're aligning with the spike proteins. And what I did as well was I took multiple mammalian enzymes or mammalian proteins that were putatively the same, and then I also align those with the spike protein, and that way, hopefully, if there is homology, that it would be both cross-species and then also cross viruses as well. So I was able to see some sequence homology that way. However, based on previous experience with the pufferfish, Takifugu rubripae proteome, and its unique evolutionary history, we restricted the search to this species. Okay, interestingly, a query using the full-length SARS-CoV-2 spike protein revealed a sole hit with the pufferfish Hepcidin protein. Given that SARS-like coronaviruses can be found in bats, we also use a full-length bat coronavirus sequence as the query, which showed a closer match with pufferfish Hepcidin. So they didn't just use the human coronavirus or the human SARS-CoV-2 coronavirus. They also used the closely related bat coronavirus to see if that was even closer to this Hepcidin. And as a query, which showed a closer match with the pufferfish Hepcidin, so it worked even better. The scores and e-values here are not meant to indicate any claims of statistical significance, but rather are provided for the purpose of comparison. So the similarity between the spike protein and the hepcidin appears to be at the cytoplasmic tail of the spike protein, or perhaps at the junction between the transmembrane and cytosolic tail domains. Multiple sequence alignment of this sequence region generated using the AlignX feature of the Vector NTI Advance 11.0. Just know that this is some program, probably a website, using 3 coronavirus spike proteins and 4 hepcidin proteins from pufferfish, bat, and humans. Pufferfish, bat, and human is illustrated in Figure 1A. So, where is it? I believe it's at the bottom. Yeah. So we'll look at this later when we finish reading. The alignment depicts a number of conserved motifs, particularly between the first pufferfish hepcidin sequence. Various pufferfish species have at least 2 hepcidin-like genes, so that's one thing that you have to keep in mind, that you are aligning the correct gene or the correct protein, and the coronavirus spike proteins. In a sense, the pufferfish sequence seems to act as a bridge between the coronavirus motifs and those in the human hepcidin sequence. The similar cysteine-rich motif takes the following form, and this is the form, so just know that the C's are cysteines, K is lysine, G is glycine, and then X is just a random amino acid. And then R and K, so this is a basic amino acid or a positive amino acid, and then phenylalanine. So just keep, you don't need to, you don't need to memorize all this. Just know that this is, I accidentally stopped the recording. 1 second. Sorry about that. So you don't need to remember the sequence. You just need to remember that it is, this is a common sequence between the pufferfish hepcidins and the coronavirus spikes, I should say. Of note, the 8 cysteines of the mature human hepcidin in the similarity motif and the aligned cysteines of the SARS-CoV-2 spike protein are not all specifically coded by one of the 2 cysteine coding codons. Both codons are present in the respective gene segments. Also, for comparison purposes, as a coronavirus envelope protein contains a related motif. This sequence was also added as the last line in the alignment. So something that I need to make note of before we read too far into this is that for some peptides, a lot of them are very cysteine rich, and the reason why is cysteines can, so cysteine is an amino acid that has a sulfur group on it, or hydroxy sulfur group. I shouldn't say hydroxy, but a sulfhydryl group on it, or a thiol group is another name for it. And these thiols are able to interact with each other and make a strong covalent bond, which is rare among the amino acids. So they can fuse, and when they fuse, there's a lot less wiggle room as far as what structures the peptide is able to do. So there are a lot of structural motifs that are very cysteine rich. So I'm thinking of cysteine knot proteins right now if you want to look into those. But just know that when you have a cysteine rich region, they could be similar not because of they might be similar not because they are imitating each other. They could just be similar because that is a very stable motif, or that is a very stable amino acid sequence where as long as the cysteines and a few key residues are the same, you can actually change a lot of the stuff around the cysteines and make completely different products, or have completely different, what would the word be, functions in biology. Continuing on, this is a distant and limited sequence similarity, and no claims about homology and sequence conservation can be made at present. So, nevertheless, this raises a potential and intriguing question of whether there could be mimicry of human hepcidin, structural or otherwise, perhaps inside the host cell by the transmembrane cytosol junction of the spike protein. It should be noted that it is unlikely for the sequence similarity reported here to be due to the relatively cysteine-rich composition of hepcidins. When searching all available teleost proteomes using the aforementioned bat coronavirus sequence, hepcidins are the only hits. So it's only hepcidins that they're coming up with, even though they are clearly not the only cysteine-rich proteins. Moreover, in addition to various hepcidin orthologs containing 8 cysteines, 4 cysteine variants have been described in nothothanoid fish species. So I'm not exactly sure what that is, but it's just a fish species. So here they're saying that there are a lot of different cysteine-rich proteins and peptides. Yet this particular sequence in this spike protein is not lining up well. It is not, there's not a lot of good cysteine homology between this spike protein piece and the other cysteine-rich proteins that are found in the human body. Granted, one of the reasons that this might be is that sometimes you might create one structural motif that is very important, and you use that structural motif for its function, and you don't want to have use it elsewhere in the body where it could interrupt the original purpose of the original hepcidin in this particular case. So there would be at least some reason why you wouldn't want to use the same very important structural motif for multiple peptides. If you wanted there to be very little crossover. So to give an example, to give a good example would be, it's not a peptide hormone, but the steroid hormones. So a lot of steroid hormones have extremely similar structure. They're all based off cholesterol, and they're all very long, relatively flat, and hydrophobic. And a lot of them have they're a little promiscuous with what enzymes and receptors they're able to interact with. So, but your body recognizes that as it's producing these hormones, that there is going to be a little bit of promiscuity between the hormone and what receptors it's going into. So, at least in the case, that would be a counterexample where you have uh, a structural motif of a hormone that is repeated in a lot of different hormones, and then they do some bleed through activity. Now, um, in the case of hepcidin, one would, I guess, argue that, okay, if you did have multiple proteins that use the same structural motif that you see here, they would probably be all doing very similar functions or would be working upon the same metabolic pathway. So, for instance, all the steroid hormones, like a lot of them work on very similar pathways. So, but when it comes to iron metabolism, don't quote me on this, but there's probably not as much variety in the iron metabolism as there is in what the steroid hormones do in the body. That seems to be a very diverse, what do you call it? There's a diverse set of metabolic pathways that those steroid hormones take, but with hepcidin, it seems to be very, at least relatively one track. So continuing, where are we at? It should be noted, moreover, in addition to various hepcidin orthologs containing 8 cysteines, 4 cysteine variants have been described in, a different type of fish, that's right. So hepcidin is a small peptide hormone that was discovered in 2000-2001 and initially named LEAP1, so liver-expressing antimicrobial peptide. So that is where the hep means liver and cidin, it kills. So it is able to kill something. So hepcidin is the liver-expressed antimicrobial peptide. So it has an anti-parallel beta sheet fold. I don't want to go too into detail, but beta sheets are a type of structural motif, secondary structure. So you can have your alpha helices, where your amino acid sequence really spirals around, and then you can do beta sheets, where the amino acid sequences are straight, but you can have the amino acid sequence go this way, and then another one go the other way right next to it. And that would be, what was that, anti-parallel? Let me just double check it. Yeah, so that would be anti-parallel beta sheet folds. And I believe, is there a parallel one? I believe there's also a parallel one, but don't quote me on that. I have to look at that. Continuing, and is involved in iron trafficking and the host's response to infection. In fact, it has been remarked by a number of commentators that hepcidin is to iron what insulin is to glucose. Clear hepcidin orthologs appear to be missing in birds and invertebrates. So clear, they might still have a hepcidin ortholog or a hepcidin version in birds and invertebrates, but it's not clear. We haven't identified it yet. The human hepcidin, coded by the HAMP gene, is an 84 amino acid pre-propeptide leading to a mature 25 amino acid peptide that is detectable in blood and urine. The pro-protein convertase furin has been demonstrated to cleave the prohepcidin at a polybasic site. So this is important. The pro-protein convertase furin has been demonstrated to cleave the prohepcidin at a polybasic site. So you start off with your giant protein, and by giant I mean the 84 amino acid pre-propeptide, and then this ends up getting cleaved into a mature 25 amino acid peptide. And one of the proteins that is known to do this cleavage is the furin, and it uses a furin-like cleavage site, which that is normally arginine-arginine, alanine-arginine. But obviously, it's very promiscuous in what type of polybasic sites it is able to attack. So in the case of hepcidin, it's actually 4 arginines. And it has recently been reported to exist in the ectodomain of SARS-CoV-2 spike protein. So they're making mention here that the furin-like cleavage site is present on the spike protein at a completely different spot. It's actually done between the S1 and the S2 subunits, but which is near the middle of the spike protein. But this cytosolic tail is near the very end of the spike protein. So just keep that in mind. Which is absent in coronaviruses of the same clade. So it's absent in coronaviruses of the same clade, but this furin-like cleavage site is actually in other coronaviruses. But not in the very closely related coronaviruses. So, granted, there is a lot of, what would the word be, selective pressure to generate or remove a furin cleavage site. There is selective pressure to do that. So just because you see a random furin cleavage site randomly appearing, just know that especially for diseases that jump species, that is a very common type of mutation done through slippage of the, the RNA-dependent RNA polymerase, which is the duplicase of the virus's RNA that can slip easily, causing that change. Let me continue. So hepcidin binds to and mediates the, let me just double check. Okay, the protein coding part of the HAMP gene is split over 3 exons with the 25 amino acid mature peptide occurring in the last exon. In terms of the putative functions, Prentice notes that although the hepcidin molecule does itself possess some antimicrobial activity, it is rather weak compared to peptides such as defensins, and its primary contribution to innate immunity is through regulation of iron. So they're saying here that this protein, or this peptide, does cause in of itself some antimicrobial activity, but that's not the just the direct antimicrobial activity is not the most important function of it. The most important function in fighting diseases is through iron metabolism. So hepcidin binds to and mediates the degradation of ferroportin, the only known cellular iron exporter. The structural details of this interaction are being mapped and studied in ever more detail. So hepcidin binds to and this ferroportin, which is a cellular iron exporter. So it is a protein which I believe is on the cell membrane, and it is spitting out the iron from inside of the cell, if I'm understanding this correctly. And then somehow hepcidin can bind to that and cause its degradation. And by causing its degradation, one would assume that that would cause a buildup of iron inside of the cell. Granted, I'm not too familiar with this. Just know that that's my takeaway from this. So if the hepcidin binds to the ferroportin and destroys it, then that means iron is going to start building up in the cell. So that would make sense, because if you're treating hepcidin to iron, what insulin does for glucose, hepcidin, if you have it expressed in the body, it's going to have the cells starting to collect or concentrate iron within the cell. So the same thing goes with insulin for sugar in your body. If you have insulin, then that's going to cause glucose to become concentrated into the cells of your body rather than existing in the in your blood serum or your plasma. So there are a number of solved structures of hepcidin. An NMR structure of human hepcidin is depicted there, including the locations of 4 putative disulfide bonds. Available solved structures of the coronavirus spike glycoprotein, as far as our search could reveal, mostly utilize expression constructs that stop just short of the transmembrane domain. So the studies of the structure of the spikes a lot of times are only focusing on the part of the spike protein that is sticking out of um, the cell or the cell or the virus membrane. So they're not as interested as the part within the membrane and the cytosolic tail that is within the cytosol or within the cell. As noted, this is partly because the protein's ectodomain is the main focus of studies on viral binding to host surface receptors. For example, these people have recently reported the cryo-EM structure of the ectodomain residues 1 to 1,208 of the spike protein, and they were able to make the trimer in the pre-fusion conformation. So I did another video going over this particular paper. They didn't, when they tried to find the structure of the spike protein, they didn't do any, they just did a genetic engineering feat where they took the outside part of the spike protein and then they were actually able to genetically engineer what's it called, a peptide at the bottom of it, which facilitates in trimerization or getting 3 of them to get very close together. And then the native structure of the spike protein is that they're trimers of 3 of the spike proteins that come together to make the final spike. Now, so that is useful for finding the structure of the outer part, because the larger the molecule you try to discover the structure of, it gets much harder the longer or the much bigger and the floppier the protein is. So rather than focus on the entire protein, the entire spike protein, they'll just focus on the ectodomain and find a way to recreate the ectodomain. But this excludes the transmembrane and the cytosolic tail domains. It goes without saying that the inclusion of the transmembrane domains would require complicated structural elucidation protocols, and even then, one may still not be able to solve the structure of the protein in its entirety. So they're saying here that it's difficult to do. So that's why they didn't do it. They just went with the easy route, especially when things are going as fast as they are right now. You would want to spit out a paper as quickly as possible. So, yeah, moreover, we would like to report that using the PFAM-A structural domain database, and this is just a database, in the HHPRED, HHPRED Remote Homology and Structure Prediction Toolkit. I assume this is just a program that you're able to put in amino acid sequences in and have some prediction on what type of structure you're going to get out of it. So remember, you have amino acids and they fold among themselves to make the actual structure of the protein. Well, there's a lot of research that goes into, if you know the amino acid sequence, rather than having to discover the structure yourself using like X-ray crystallography or some other protocol, if you could have a program where you just type in the amino acid sequence and then it predicts what structures are likely, a lot of research has gone into that particular field. So this is a structure domain database that they are going into to try to elucidate some information from the spike's amino acid sequence. Anyways, the coronavirus spike protein regions are analyzed here, show some predicted structural similarity to the lipolysis-stimulated receptor, or the LSR lipoprotein receptor, and Hepcidin sequence show some predicted structural similarity to the SAR8.2 protein family found in the Solanaceae plants. What, if any, significance these findings may hold is unclear at present. So they just included it. I don't think they made any more statements about that. Of more importance right now would be the theoretical and/or actual elucidation of the structure of the spike protein, transmembrane, cytosolic tail junction region, and a comparison with the available hepcidin structures. So given the prominence of cysteines in the aligned motif, how are they utilized in the respective similar domains? At first pass, the usage appears to be different. As noted earlier, in hepcidin, the cysteines may give rise to a compact disulfide-bridged peptides, whereas in coronavirus spike glycoproteins, the cysteines in the transmembrane cytosolic tail junctions serve as palmitylation acceptor residues. So these cysteines in the so in hepcidin, the cysteines are putatively attached to each other through those disulfide linkages. But in the SARS coronavirus, these cysteines are actually covalently bound to palmitoleic acid. So I'll get a photo up right now. So basically, palmitol, think of palm oil. There's a lot of there. Oh, didn't mean to go there. There is a lot of oil in palm, right? So palm oil. How to phrase that? Some of the fatty acids in palm oil were actually named after the palm based on the number of carbons that are in it and the fact that it's saturated. So when you think of palmitolization, think of you're adding a very long fatty acid onto a chemical in order to make it more hydrophobic. And when you make it more hydrophobic, then it'll be able to go interact with the cellular membrane more. So remember, the cellular membrane has the polar ends on the outside of the membrane, and then it has the fatty inside of it. And that fatty inside will interact with this fatty portion of the palmitic acid. So yeah, so here you see the fatty acid. So this is a acid group, carboxylic acid, so like vinegar. And then rather than being vinegar, though, it's a very long carbon chain, and this is hydrophobic. So, and then it's also named after palm, if I remember correctly. So, and here you can see where you have this was a cysteine. So here's your protein amino acids side chain or chain, and here's your cysteine residue. So this sulfur group in the cysteine ends up getting palmitoylated, in which they add the palmitoyl group to it, and this would make the cysteine more, or this would make the protein hydrophobic in this region. Okay. So here, the cysteines serve as palmitylation sites of the spike protein that facilitate membrane fusion. And at least a portion of the palmitylation of the SARS-CoV spike protein has been reported to occur in the premedial Golgi compartment. So this means that the palmitylation is happening early, or it's happening before the virus is even fully produced. So, however, there is also the possibility of cross-disulfide bond formation with a non-homologous small cluster of cysteines within the envelope protein. Moreover, if S-palmitylation is reversible and a dynamic process, it is to be determined if the spike protein junction cysteines might in fact have a different post-translational modification in the host cytoplasmic environment, although there is no evidence of this in the moment. So what they're saying is, well, a lot of proteins, they're dynamic in what structure they have and what modifications happen to them. So for instance, you can do things like glycosylation. So you can have N-linked and O-linked glycosylation where you're adding sugars to your protein. And that happens on the surface of the proteins, and then that helps with the shuttling of the proteins throughout its maturation. You can have things like palmitylation. You can have things like proteolytic cleavage, where you break the protein into multiple pieces into their final active forms. So the modifications that happen to proteins is sometimes what word they use specifically? different. Oh, dynamic. So they said it could be a dynamic process where things are changing all the time, and maybe there could be a post-translational modification that you do at one point in the life cycle of the virus that could be reversed at a different part of the life cycle of the virus. We do not know this yet for the spike protein. Continuing. In the cited paper by McBride and McHammer, the authors conclude that the palmatization of the SARS-CoV-2 spike protein was not necessary for the S protein stability, trafficking, or subcellular localization, nor for the efficient interaction with the M protein. So for the original SARS, the original SARS had these cysteines as well, and there had been tests done on them to see whether or not this palmatization was necessary for the SARS virus to function properly, and it wasn't necessary. It was not necessary, although it might have helped, but it was not necessary for the protein stability, trafficking, or subcellular localization. To what extent the post-translational modifications of the spike protein and hepcidin, be it furin cleavage, disulfide bonds, or palmatization, are in any way similar to the intracellular context remains an open question. In terms of the possibility of an evolutionary connection between the spike protein and hepcidin, one could imagine a scenario where an ancestral spike protein acquired the hepcidin-like sequence from a host organism, and the new sequence was palmitoylated to aid with membrane association. Lee points out that the primordial form of the coronavirus spikes might contain the S2 only, or the second portion of the spike protein, which is where you get the transmembrane and the what would you call it, cytoplasmic tail domains. That is all within the S2. Granted, the receptor binding domain for SARS-CoV-2 is in the S1 region. So this Lee would probably argue that the original viruses did not have the same receptor binding as what it has evolved to later. So basically, Lee is saying that the S2 is probably the primordial form, and then over time, the S1 came, and then obviously after the S1 came, it was evolutionarily advantageous in some way, shape, or form. So these people are arguing that maybe in the past, the earliest coronaviruses or coronavirus-like things could have possibly taken a gene of hepcidin or a part of the hepcidin gene and then used it for its own purposes, which is a perfectly rational explanation, where viruses are going into cells all the time, and the cells have different types of bits of RNA that are floating around, and sometimes there can be some homology, or not homology, what would the word be? Complementarity of the RNA that's just floating around in the cell and the RNA that is a part of the virus genetics. And sometimes, if those line up and you get the replicase machinery attaching to that, there could be a way to splice bits of RNA together. It is possible. Regardless, continuing, and the cytoplasmic motif features highlighted in the current report do not appear to be present in other class 1 viral membrane fusion proteins, which include the influenza virus. So they looked to see if this motif or the structure was in other viruses, and it's pretty exclusive to coronaviruses, they're saying. Although we have not performed an exhaustive search, however, a number of questions might arise under such a scenario, such as the difference between the primary localizations of hepcidin, considering its putative interaction with extracellular and transmembrane regions of ferroportin, versus the cytoplasmic domain of the spike protein. Alternatively, it might be argued that perhaps a case of convergent sequence evolution is at play. So they're saying it might not have been the acquiring of hepcidin. It could just be that part of the coronaviruses started mutating these motifs all by themselves, and as time got on, it got closer and closer to doing its function by either inhibiting or acting like hepcidin, and that was evolutionarily conserved. But they're saying this is something that happened in the coronaviruses. It's not something that you see in other viruses. For example, the influenza virus hemagglutinin glycoprotein appears to have a conserved CXI-CI motif in its cytoplasmic tail domain, and perhaps an ancestral spike protein with similar features convergently acquired hepcidin-like sequence motifs. These are of course, speculations and remain open questions. Investigations pursuing these topics could also make use of studies that attempt to trace the evolutionary history of hepcidin itself. So given the central role of hepcidin in iron metabolism, it is important to point out a number of circumstantial but perhaps important findings as pertaining to the topic at hand in the literature. These include a link between SARS and liver function abnormalities, the association of pulmonary iron overload and restrictive lung disease, the role of iron in pulmonary fibrosis, and hepcidin's modulation of the proliferation in pulmonary artery smooth muscle cells. Furthermore, using experiments of iron overload in European bass, Neves and colleagues discuss the functional partnership between hepcidin and ferroportin from an evolutionary perspective and suggest that this may open new possibilities of the pharmaceutical use of selected fish. Hepcidins during infections with no impact on iron homeostasis. Oh, open new possibilities, use of selected fish, hepcidins during infections with no okay. Oh, wow. Okay, so I just needed a second to process that. So basically, there was another group that had made the suggestion that you could be using the hepcidins from fish, from specific fish, and use them as pharmaceuticals because they don't interfere with your iron homeostasis, but could still be serving as a pharmaceutical. Yeah, I'll have to look into that a little bit. Lastly, hepcidin upregulation along with serum iron reduction has been reported in influenza infections. Oh, okay, I did not know that. So, yeah, basically, if you have an influenza infection, somehow the influenza is encouraging the iron to rather be in your plasma to go into the cells. And that would presumably have an effect on the virus's ability to duplicate and function and be more virulent, which may possibly suggest that a potential spike protein hepcidin mimicry could allow the SARS-CoV-2 virus to exacerbate this response. Importantly, however, iron dysregulation changes may only be at local cellular tissue level and not reach a systemic response. Maybe, but if you see virus all over the body, then it is possible that you're going to have a systemic response affecting your entire system. Specific to COVID-19, a number of broader questions that could follow up from this work are, first, does the spike protein, similar to hepcidin, potentially promote iron sequestration in alveolar macrophages, and hence impede the host's immunological response? Second, could a recent report of the common presence of digestive symptoms in COVID-19 patients be explainable in part by the link to hepcidin? And third, could systemic changes in serum iron levels, with a possible view on the degree of above normal serum ferritin in patients, so this is another, especially out of the post-mortem results, the ferritin levels of the people who had died of COVID-19 in China had higher ferritin levels, which would indicate that they had more, that there was something going on with iron metabolism. Or levels of hepcidin itself be detected in patients with varying COVID-19 severities. So Figure 1, comparison of select hepcidin and coronavirus spike proteins. Um, we'll They didn't do a good job aligning this. So here we have a sequence alignment of the different coronaviruses. So here we have MERS, SARS-CoV-2, and a BAT-CoV spike, and this is the cytoplasmic tail region, as you can see here. So as I said earlier, you have the S1, which contains the receptor binding domain, and then you have the S2 region, which will have the transmembrane domain, and it also has the transmembrane domain and the cytoplasmic tail. And then, yeah, they're calling it the membrane fusion subunit. In this case, I've just referred to it as the S sub 2. So here you can see human hepcidin. And then, let's see, I believe this is the human hepcidin. So this is the amino acid sequence of the human hepcidin. And here you have your polyarginine tail, or polyarginine furin-like cleavage site. And this polyarginine furin-like cleavage site will split here at this D. Well, okay, they're split there at that D, and then this is the hepcidin molecule that you see here. And here you have the disulfide linkages, so you'll have 1, 2, 3, 4, 5, 6, 7, 8 cysteines in here, and then each of these have a putative disulfide linkage going on between them. And this is one of the beta sheets, and then this is the antiparallel beta sheet. And so this is the structure of hepcidin. It's floating around in your body, and it's influencing your iron metabolism. So they've done a sequence alignment with multiple coronaviruses and then multiple hepcidin molecules, and there is good homology, according to them, between the cytoplasmic domain of the spike proteins and the hepcidins of different species. And here they compared the SARS-CoV-2 spike cytoplasmic tail right here with the human hepcidin. And so for the human hepcidin, you have the disulfide linkages described here with these SS's. And for the SARS-CoV-2 spike protein, they have the palmitylization described by these palmitoyl groups to the cysteines over here. Okay, and so what they argued was that it might be a dynamic process where maybe this palmitylization happens and then stops, like it stops happening, or I shouldn't say stops, but it gets cleaved off later. So if it's a dynamic process, maybe it can go from this into a structure that is more similar to this. So what I did was I also wanted to do a similar alignment. So here I have the spikes of the spike SARS-CoV-2, the original SARS spike, and then what was it? And then these are two. This one's a bat coronavirus, and this is just another similarly related coronavirus. And here I have the hepcidin peptide in purple. So this is where the hepcidin peptide is. So here you can see hep C, it's the bottom one. And then here is the polybasic furin cleavage site that the furin will latch onto and then cleave right after it. And you can see here, there is pretty good homology. Most of it is, well, so you have a threonine. Here you have all hydrophobic, what do you call it? Amino acid sequences. So in spikes SARS-CoV-2, it's a methionine. In the other spikes, they're leucines, and then in hepcidin, it's a phenylalanine. You got another cysteine, alignment of cysteines, and then you have enough methionine instead of isoleucine, which is also hydrophobic. More cysteines, cysteine, and then here you have a positively charged amino acid sequence, more cysteines, and you know the drill. So this is in the cytoplasmic tail. So if this is inside of the cell, it's not outside, it's not floating around freely, how could this possibly have any effect on hepcidin? Well, I can think of multiple ways that it could influence hepcidin regulation. So, for one, if it looks similarly enough, and if you are able to generate an antibody against it, then those antibodies might be able to interact with hepcidin. Now, granted, this is in the cytoplasmic tail. It's within the virus. It's within the cell, so it's not exposed to your immune system as the full functioning protein. It's not exposed. Now, that doesn't necessarily say whether or not this ends up getting cleaved in some way, shape, or form. So I'm looking through, I'm trying to find, let me look at the positive residues. So, I mean, I'm not seeing that much that would indicate that it's a very an easy to cleave location, but theoretically, if a protease was able to come through here and cleave a part of this randomly, either purposefully purposefully of the virus, maybe it wants it to be cleaved, or another possibility is just since the production of the viral proteins is a very messy process, especially near the end of it when the cell literally just explodes before all the virus particles have assembled. It could be that the cytoplasmic tail is actually exposed to the immune cells in specific conditions. So there is that. The other possibility is that it could influence hepcidin regulation by either imitating it to activate receptors, or it could imitate it to deactivate receptors. So just because something looks similar doesn't mean that it's going to be doing the same function. It could actually be inhibiting the function or doing a good job at activating the function. So just because things look similar doesn't mean that you can say that they serve the same function. So trying to think of an example. So for instance, testosterone and, oh no, that's not a good one. I can't think of anything on top of my head. Just know that some medications, for instance, will create a compound that looks similar to a chemical that is in your own body. And sometimes those chemicals can do an even better job at interacting with the receptors in your body, and thus are improving or eliciting that response. And sometimes they do the opposite, where they will bind and then not do anything. And so that would be, and then that would lower the effect of that hormone in your body. So that would be the difference between an antagonist and an agonist. But that is besides the point. What we're saying here is that here we have a sequence that is similar and conserved, at least in some coronavirus spikes, that is very, very similar to, I mean, very similar is relative, right? I mean, to me, it's not very similar, but the cysteines are similar, and they're similar to the hepcidin molecule. So would this adopt a similar shape? Probably. It would probably adopt a similar shape if the palmatization ended up coming off at some point, or maybe some of it never gets put on. So it is possible that in the reproduction of the virus, near the end of the life cycle, you might still have a lot of viral proteins that weren't able to fully get into viruses. And they weren't able to get all the post-translational modifications needed to go into a virus. So you could have just these parts floating around in the cell, and then the cell dies and then releases it into the environment before it can fully become part of a virus. That is something to consider. Another thing to consider is the virus replication machinery is sometimes incomplete in its ability to duplicate an entire part of the RNA. So if it only duplicates part of the RNA, it is possible that you end up with parts or pieces of the fully functioning protein. You might just end up with pieces of it. So that is another way that you could just develop this portion, or at least part of this spike protein, rather than the full functioning thing. Another thing is the protease activity could be breaking up the spike proteins into these smaller peptides. So those are just some ways that you can get this particular sequence floating around, being able to act at least somewhat like a hormone. Now, does this behave exactly like the hepcidin, or is it behaving like the opposite of hepcidin, or is it behaving in a way to generate antibodies against hepcidin? There really is a laundry list of different things that could be happening. So if you could imagine, if you started making antibodies against hepcidin, then, well, I mean, let's brainstorm that for a little bit. So let's say at the very beginning, when the virus is wanting to duplicate a lot, and the virus wants to get iron into the cell, maybe it wants to have these hepcidin-like pieces floating around, because then that helps shuttle iron into the cell. And that'd be good for a virus that is wanting to make sure it has enough metal cofactors for its proteins. But then at the end of your illness, or when the virus has fully taken hold of the body and then wants to spread, maybe it wants to generate some pathology. So in that case, you might be generating antibodies for this particular sequence. And then when you make antibodies for that sequence, those antibodies might cross-react with the hepcidin that is actually floating around in your body. And when that happens, the iron, rather than going into your cells, just stays in your blood. And that could have some very serious implications, such as blood clotting would be one of the possible symptoms of having excess iron in your blood. Another one would be the production of free radicals and the upregulation of the inflammatory response. Don't quote me on that. I have to double check to see if that is accurate, if that is actually what would happen. But if you just had a bunch of free iron floating around in your system, that could be bad as well. So what else do I want to talk about? Oops, not there. So yeah, just keep that in mind. That is the thesis of these people. They are arguing, or this person is arguing, we probably should put some attention into this, because it is possible that this sequence it's definitely being used by the virus for some function. It definitely, whether that be structural or as a function in its pathology or reproducibility, it's there and it's conserved, and so it's doing something. Whether that something is something that we can develop treatments for, or if that something is predictive of what type of symptoms someone has, then it could be a target for how we can improve the lives of people who do, in fact, contract this virus. So with that, oh, I do want to mention before all this that this is just a brief communication, and this is still on archive, so it's not, it hasn't been peer reviewed yet, and I don't know if it will be peer reviewed, but it hasn't been peer reviewed yet. But they're not making any real definitive statements other than we should look into this. So it just stuck out to me because I also had a similar epiphany over the fact that iron regulation is probably playing a very large role in the duplication of these particular viruses. Now, what would concern me? yeah, we did this for a while, but what would concern me specifically is if we start making antibodies against this virus and we are not very selective at which antibodies we produce. So if you get a vaccine, sometimes you can really make sure that the vaccine is not having you produce antibodies that create long-term negative consequences. When you are exposed to just a straight up virus, though, your immune system can negatively be, or at least permanently be altered in a way that is damaging for you. So if you could imagine, if you are, if you have created antibodies against this viral epitope and it cross reacts with the hepcidin that is in your body, you won't be able to regulate your iron levels as effectively as you had in the past. And that might be a permanent thing for certain people, where some people will never be able to regulate their iron effectively for the rest of their life. Possibly. That is just speculation, but there could be a subset of people where that actually happens too. And the problem with autoimmune diseases is that they're very hard to stop. You once you have it, there would be a lot of. I need to brush up on what medications you would technically use for this. Just know that it would be very difficult to ramp down your immune response to your own body. So if you run through that scenario where the hepcidin is not being freely released, or the hepcidin is not being able to roam freely throughout your own body, it keeps on getting sequestered by antibodies. That means that it'd be similar to having insulin resistance, but with iron. You might have high blood iron, and you would have a hard time managing your blood iron and making sure that the blood iron gets sequestered into your cells or gets sequestered into the appropriate proteins that prevent them from causing oxidative damage and causing blood clotting. That is something that we really need to consider. Especially, so I should say the other coronaviruses do have sequences that are somewhat similar like this. So the less virulent coronaviruses, a lot of them actually, yeah, so I did the same type of alignment with some other coronaviruses. They are cysteine rich, but not all of them align nearly as well as the SARS-CoV-2 and the SARS coronavirus. So this is a different spike protein of another one of the common cold coronaviruses. This one is NL, which I believe is one of the ones that utilizes the ACE2 receptor to get into the cell, and they can actually, their spike protein can line up pretty darn well, where they can have a series of 5 amino acids that line up perfectly, and then a few other cysteines down there. So this is partially conserved, but why do we not see the same effects, or why do we not have as much iron dysregulation when it comes to the common cold coronaviruses? So what I would argue is, well, how do we know? Like, how do we know that these other coronaviruses aren't also causing some level of iron dysregulation in the body? So, I mean, to a degree, they probably, if this particular hypothesis is correct for the SARS, SARS-CoV-2, and the MERS coronavirus, where they cause significant iron dysregulation, well, maybe the other coronaviruses are also doing significant iron dysregulation as well. I mean, we haven't ruled it out. But where the issue would be worse would be the fact that SARS, SARS-CoV-2, and MERS are much more virulent, and they are producing a lot more of these peptides. So since they are so virulent, and since they can duplicate so much in the body, and at least for SARS-CoV-2, it is able to infect a lot of cells in your body, not just being restricted to the lungs. So because of that, you could have a full body response to these peptides if they do have some physiological purpose. And so whatever they're doing, it's like you're getting inoculated, or it's like you're getting a bigger load of whatever this peptide does to the body. And with the common cold coronaviruses, maybe if you had a lot of that same peptide, it would affect you just as much. But since your immune system can usually take care of the common cold very, very effectively, it doesn't become that serious. With these coronavirus, with SARS-CoV-2 and MERS, that might not necessarily be the case. We might see a lot more long-term side effects, especially if they are immune system or autoantibody related. You can see those well into the future. So one of the concerns I have is something with beta endorphin and the cause of depression and psychiatric disorders in a lot of people who survived from the first SARS. So I believe out of all of the survivors, and if you take out the people who had psychiatric disorders before they even got diagnosed with SARS, about half of them had a psychiatric disorder a year later. Now, most of it was PTSD, but like there are serious, and a lot of them also had a family member or a close or loved one who had died from SARS, so there might be some survivor's guilt as well. But my concern is there's also a biophysical aspect to it as well. Are people developing autoimmunities to some of the important hormones in the body, like prolactin or prolactin release? What would the word be? Or at least peptides that inhibit the release of prolactin, which I went over in a previous video. That would be a reason of concern, especially when it comes to the reproductive and the steroid system. And then there's also the beta endorphins that I'm worried about, where if you create autoimmunity for the beta endorphins, then that's going to cause depressive-like symptoms as well, or at least a dysregulation, in the opioid response. So there's a lot of things that I worry about when you get a high dose or an acute dosage of these peptides, because these peptides are theoretically trying to influence your metabolism in one way, shape, or form. And if you get a large load of a metabolically active substance, then the effects might be long-term. So it's not just the acute things you need to worry about. You need to worry about the chronic effects, and the chronic effects could be worse from a worse coronavirus versus a more common cold coronavirus. So with that, I'm going to leave, and thanks for watching. If you have any questions at all, leave them into this description. All 4 of my subscribers, so I had 3 of you from a long time ago. So yeah, so leave them down, and if there are any topics that you want me to go over as well, leave those in the description. And if you found any mistakes, or if I misspoke anything at all, remind me so that I can take care of that in the next video. But with that, thanks for everything.